Reporting for Group 17, I'm Rachel.
I'm Morgan.
I'm Ashley.
I'm Ming.
My name is Rachel, and thank you for joining Breaking News on Channel 17. We're investigating the discovery of cattle genetics and the microbial population influence. With us now is ASI 500 genetics student Morgan Gantz. Can you tell us a little bit about the article that you have read?
The rumen is the largest of 4 compartments in a cow's stomach and the place where microbial organisms are involved in digestion. The genetic makeup and diversity of rumen microbes in a cow's stomach is critical to the optimum nutrient digestion. Unlike humans, in which acid and enzymatic digestion is the first process to take place in the human digestive tract, microbial digestion is the first process to take place in feeds and forages a cow consumes, in the first line of digestion. By modulating the microbial composition of the rumen, we may ultimately be able to select cows with a rumen microbial population more adept and efficient at using high-energy diets.
Thank you. That sounds fascinating. To look into this more, also joining us today is ASI 500 genetics student Min. Min, tell everyone following this discovery, how did you find out about the microbial composition of individual cows?
Okay, so basically we extract DNA from samples and amplify 16SRA gene using universal PCR primers, which match highly conserved regions of 16SRA gene. And 16SRA gene also contain hypervariable region, which would provide species-specific signature for us to identify the origin of organism. In that case, we define who's there without need to culture.
We are using the beads in this container as representation of the microbial population in an individual cow. As you can see, I'm removing a section of beads from the container and extracting the DNA from those beads. We need to purify the DNA and using PCR to amplify the 16SRA gene. Then we use next-generation sequencing technique to sequence the PCR product and compare the sequence we got with the database, so that we can identify the microbial species within the rumen microbiome.
Channel 17 decided to get a further look into the 16 rRNA gene in the microbiome that takes place on a personal level with a cow.
So let's look at this big picture. How does this all relate? So in this model demonstration of a tub as the rumen with both microbes, there is a normal amount of good microbes and bad microbes that live there together in a community without causing problems. But when a cow eats contaminated food, as shown in the skip by M&Ms going into the cow's food, whether it be corn that has not been fermented properly or feed contaminated with black mold, it begins to form an ideal environment for those bad microbes to thrive. These bad microbes begin to overpower the rumen by taking essential nutrients from good microbes, causing them to die, which is ultimately how a cow gets sick. Here you can see the combination of too many bad microbes in the rumen, taking the nutrient which is killing off good microbes. When we extract the 16S rRNA gene from inside a cow's rumen, we use the concept of metagenomics to determine the levels of good and bad bacteria or microbes that are in the rumen when a cow is healthy, and then compare this to the levels of a sick cow to figure out why the cow is sick.
Bottom line for people who want to take care of cows, make sure that your feed is processed and produced in a proper way, because if not, there can be serious consequences to the cow's microbiome that can cause serious health problems and could lead to death. Back to you, Jerry. Here at Channel 17, we investigate through all areas of problems and talk to everyone. 4961, how was your day today?
It's great. How are you, Rachel? I'm so excited you came to visit.
Are your microorganisms treating you well?
I think so. Are yours?
Well, mine are not the same as yours, so I will not actually understand.
Oh, you taste so good, Rachel.
Bam.